Hello everyone, welcome to JK Biology. In this video, we'll discuss about early stages of embryonic development in Drosophila. In Drosophila, the process of embryogenesis begins at fertilization, but most molecular events critical for Drosophila embryogenesis occur during oogenesis. And this is very important. This question was previously asked in the kset examination, that is, the most critical events for the Drosophila embryogenesis occur during which stage? The most molecular events which are required or critical for the Drosophila embryogenesis occur during the oogenesis stage. During oogenesis, the oocyte is generated by a series of cell division. This oocyte will be produced by a series of cell division. The oogonium. What is this oogonium? Oogonium is nothing but germ cell which produce oocyte is called as the oogonium. Oogonium, that is nothing but the germ cell which produce the oocyte, is called as the oogonium. This oogonium divides 4 times to produce the 16 cells. The oogonium undergo division 4 times to produce the 16 cells. Among these 16 cells, one cell will become oocyte and others become nerve cells. That means there is one oocyte and nerve cells are 15 nerve cells. The oocyte and nerve cells are surrounded by ovarian follicle cells to form egg chamber. Remember, egg chamber means that is having the oocyte plus nerve cells. Oocyte and nerve cells. Egg chamber means it is having oocyte and nerve cells. These two are covered by ovarian follicle cells. This structure is nothing but the egg chamber. Just remember, egg chamber means oocyte is there, along with that nerve cells are there, and these two structures are covered by ovarian follicle cells, and this structure is called as the egg chamber. The nerve cells produce mRNAs required for the establishment of anterior-posterior polarity and dorsoventral polarity in Drosophila. The function of the nerve cells is that these nerve cells will produce the mRNAs which are involved in the recognition of the anterior-posterior polarity and dorsoventral polarity. That means the mRNAs which are required for the anterior-posterior polarity and dorsoventral polarity are produced by which cells? Nerve cells. The anterior-posterior polarity of the Drosophila embryo is established while the oocyte is still in the egg chamber. This is very important. When the anterior-posterior polarity of the Drosophila is established while the oocyte is still in the egg chamber. Here is the diagram of the Drosophila embryo during the oogenesis. See here, this complete white portion is oocyte, and these cells are nerve cells, and this layer of cell is follicle cells, ovarian follicle cells, and this complete structure, that is the nerve cells, oocyte, are covered by ovarian follicle cells, and this structure is called as the egg chamber. Is it clear? During fertilization, the sperm enter into the egg through micropyle region. Remember, during the fertilization, the sperm enter into the egg through which region? Micropyle region, which is nothing but which forms the anterior region. Micropyle which is present at the anterior region. This portion is nothing but this is the micropyle region. Through this micropyle region, the sperm will enter inside the egg. Type of cleavage in case of the Drosophila is superficial cleavage or syncytial cleavage. This is very important. Which type of cleavage is seen in case of the Drosophila? Superficial cleavage or syncytial cleavage? Now, what is the superficial cleavage or syncytial cleavage? We'll see. In Drosophila, after fertilization, the diploid nucleus undergo a series of karyokinesis without cytokinesis. What happens? See here, after the fertilization, this nucleus becomes the diploid nucleus. Now, this diploid nucleus undergoes series of karyokinesis. Karyokinesis, that is nothing but the karyo means nucleus, kinesis means division. Kinesis means division. That means division of the nucleus. Without cytoplasmic division, cytokinesis. Kinesis means division. That is cytokinesis nothing but the division of the cytoplasm. Now, after fertilization, this diploid nucleus undergo rapid divisions without cytoplasmic division. And as a result of this, there will form a structure which is called as the syncytium. Now, what is the syncytium? Syncytium is nothing but, here I will write here, single cell with many nuclei residing in common cytoplasm. This structure is called as the syncytium. Syncytium, that is nothing but a single cell with many nuclei residing in common cytoplasm. Later, most of the nuclei start migration towards the periphery and form single layer of cell called syncytial blastoderms. Here, after the much divisions, these nucleus start moving towards the terminal portion, periphery portion, and they will form, see here, they will form a single layer of cell. This structure is called as the syncytial blastoderm. This is called as syncytial blastoderm. Now, later, plasma membrane encloses each nucleus, thereby transforms syncytial blastoderm into cellular blastoderm. Later, what happens? This syncytial blastoderm get converted into cellular blastoderm. How it get converted into cellular blastoderm? We'll see here. See here, in case of the syncytial blastoderm, the nuclei, each nuclei which are present in the peripheral region, they are not completely covered by the plasma membrane, but they are partially covered. But later what happens? These each nuclei which are located in the peripheral region are completely covered by the plasma membrane. This structure is called as the cellular blastoderm. Only difference between the syncytial blastoderm and cellular blastoderm is, in case of the syncytial blastoderm, the each nuclei at the peripheral region are not completely covered by the plasma membrane. In case of the cellular blastoderm, the each nucleus which is located at the peripheral region is completely covered by the plasma membrane. Is it clear? Some of the nuclei present in the extreme posterior end forms the pole cells. See here, some of the nuclei which are present at the extreme pole cells, these will form the, see here, these cells will form the pole cells which are present at the extreme posterior end. And the function of the pole cell, they will give rise to gametes in the adult Drosophila. Next is genetic control of embryonic development in Drosophila. Embryonic development of Drosophila is controlled by the two sets of genes. Those are next one is genetic control of embryonic development in Drosophila. Embryonic development in Drosophila is controlled by two sets of genes. Those are the maternal effect genes and another one is the zygotic genes. We'll see one by one. Maternal effect genes. Maternal effect gene, as the name suggests, maternal, that is related with the mother. Maternal effect genes are expressed during the oogenesis before fertilization. Remember, maternal effect genes are expressed before the fertilization during oogenesis. That means these genes are placed into the oocyte by mother fly itself. The genes are transcribed from mother's DNA. The maternal effect genes are transcribed from the mother's DNA. The product of maternal effect genes is nothing but the maternal mRNA, and these maternal mRNA are produced by the nurse cells. Maternal effect genes help for the development of anterior posterior dorsal ventral polarity. Function of the maternal effect gene, that maternal effect genes are involved in the recognition of the anterior posterior and dorsal ventral polarity. Second type of genes, those are the zygotic genes. As the name suggests, these genes, zygote, that will form after the fertilization. Zygotic genes means these will express after the fertilization, whereas the maternal effect genes are expressed before fertilization. Zygotic genes expressed after fertilization. These genes are activated in the fertilized egg after the fertilization. These genes are transcribed from the zygotic DNA or zygote's DNA. There are further two types of zygotic genes. Those are the segmentation genes, and another one is the homeotic selector genes. Further, these segmentation genes are of three types. Those are the gap genes, parole genes, segment polarity genes. This is brief introduction of the maternal effect genes and zygotic genes. More information will be given in the next video. Thank you. If you like my video, please like, comment, subscribe and share. Thank you.